Hello viewers, today we are going to discuss about lipid oxidation, biosynthesis of fatty acids and ketosis. The objectives of this class are to enable the students to understand the reactions of lipid oxidation, to acquaint them with the process of biosynthesis of fatty acids, to familiarize them with the mechanisms of ketosis. Triacyl glycerols are the most abundant source of energy and provide energy twice as much as carbohydrates and proteins. This is achieved because the fatty acids which are present in the triacyl glycerols are already in the reduced form. The efficiency of energy storage in fats is probably an important reason why animals store most of their energy as fats and only a small amount of energy as carbohydrates. When glucose levels are low during long periods between meals, the hormone glucagon secreted by the pancreas stimulates adipose tissue lipase activity to release fatty acids from triglycerides. Epinephrine can stimulate the same activity to release energy during the fight or flight response. Once released, these fatty acids travel through the blood to other tissues such as muscle, where they are oxidized to provide energy through the mitochondrial beta oxidation pathway. Since fatty acid beta oxidation occurs in the mitochondrial matrix, long chain fatty acids must be actively transported from the cytoplasm into mitochondria by the enzyme carnitine palmityl transferase. Now, fatty acids with 4 to 8 or 6 to 12 carbon atoms in length, which are referred to as short and medium chain fatty acids. In short, they are called as SCFAs and MCFAs respectively. These are oxidized exclusively in the mitochondria. Long chain fatty acids, which are 10 to 16 carbons long, are oxidized in both the mitochondria and the peroxisomes, with the peroxisomes exhibiting preference for 14 carbon and longer fatty acids. Now, fatty acids must be first activated in the cytoplasm before being oxidized in the mitochondria. Activation is catalyzed by fatty acyl CoA synthetases, which are also called as fatty acyl CoA ligases or thiokinases. Now, you can see the reaction here. The fatty acid combines with coenzyme A in presence of the enzyme acyl CoA synthetase to form fatty acyl coenzyme A, and water molecule is released. So, this is the first activation step. Now, the transport of fatty acyl coenzyme A into the mitochondria is accomplished via an acyl carnitine intermediate, which itself is generated by the action of carnitine palmityl transferase 1, which is also called as CPT1, an enzyme that resides in the outer mitochondrial membrane. Now, fatty acyl carnitine molecules cross the inner mitochondrial membrane through carnitine acyl carnitine translocases, and these are acted on by the inner mitochondrial membrane carnitine palmityl transferase 2, which is also called as CP2, regenerating the fatty acyl coenzyme A molecules. Once inside the mitochondria, the fatty acyl coenzyme A is a substrate for the beta oxidation machinery. The whole mechanism which I have explained now, you can see here fatty acid coenzyme A ATP combines in presence of this enzyme acyl CoA synthetase to form fatty acyl coenzyme A molecule. Now, this fatty acyl coenzyme A molecule combines with the carnitine molecule in presence of the enzyme carnitine palmityl transferase 1, which is also called as CPT1. So, this enzyme catalyzes this reaction of combining these two to form fatty acyl carnitine. You can see fatty acyl coenzyme A carnitine in presence of this enzyme are converted to fatty acyl carnitine. Now, fatty acyl carnitine through another enzyme carnitine acyl carnitine translocase is sent outer mitochondrial membrane through the inner mitochondrial membrane, and it comes out to form fatty acyl carnitine again. That means, as you can see, fatty acyl carnitine, to cross this membrane we need a transport protein molecule, and this is the transport protein molecule, which is carnitine acyl carnitine translocase, this enzyme. So, fatty acyl carnitine comes out, then it combines with coenzyme A in presence of another enzyme carnitine palmityl transferase 2, which is also called as CP2, to form fatty acyl coenzyme A. So, this is the whole process of activation of fatty acids. So, fatty acids combines with coenzyme A ATP molecule to form fatty acyl coenzyme A molecule, for which carnitine plays a very important role. This is what is given as transport of fatty acids from the cytoplasm to the inner mitochondrial space for oxidation. This is the first step of beta oxidation, which is also called as activation process. Now, next we will go into the next steps of beta oxidation. The process of mitochondrial fatty acid oxidation is termed beta oxidation, since it occurs through the sequential removal of 2 carbon units by oxidation at the beta carbon position of the fatty acyl coenzyme A molecule. Each round of beta oxidation involves 4 steps that in order are oxidation, hydration, another oxidation and cleavage. Once the fatty acids have been transported to the mitochondrial matrix via carnitine pathway, beta oxidation occurs within the mitochondria in 4 steps as discussed below. Now, first step, all the steps are given in a sequence of reactions here, but we will see the reactions as it is. We can see here fatty acyl coenzyme A, which we have seen in the previous reaction, where there is activation of fatty acid is taking place to convert itself to fatty acyl coenzyme A. Now, fatty acyl coenzyme A, you can see, is acted upon by the enzyme acyl CoA dehydrogenase. That means it removes 2 hydrogen atoms. As you understand, dehydrogenase means it removes 2 hydrogen atoms. FAD acts as a cofactor. So, in presence of this dehydrogenase enzyme, 2 hydrogens from this 2 CH2s are removed, so that you can see there is a double bond here between CH and CH, there is a double bond. So, 2 hydrogens are removed and these 2 hydrogens are taken by FAD. So, FAD is converted to FADH2. Now, in the next reaction, so acyl CoA, fatty acyl CoA, which is a substrate, is acted upon by acyl CoA dehydrogenase enzyme in presence of FAD to convert itself to trans-enoyl coenzyme A. This is the first reaction of beta oxidation. Now, after this trans-enoyl CoA is formed, it is acted upon by another enzyme, enoyl CoA hydratase. Hydratase means it takes up one water molecule, and this water molecule is added up across the double bond. So, you see here there is a double bond CH, double bond CH. So, water molecule is added across the double bond, and if you see here, one CH, this CH is taking up one hydrogen atom and this CH is taking up another hydroxyl group. So, this water molecule is added up across this double bond, where this carbon atom takes up one hydrogen. So, this CH is converted to CH2. Now, this CH is converted to CHOH. So, it is called as hydroxy acyl CoA, L-hydroxy acyl CoA. Now, this substance is then acted upon by another dehydrogenase enzyme, hydroxy acyl CoA dehydrogenase enzyme, in presence of NAD molecule. So, NAD takes up 2 hydrogen atoms. So, it is converted to NADH plus H, and you can see here the reaction is taking place again here only. So, if you see here, one hydrogen from here and one hydrogen from here are removed. So, you get back CH2 and CO. So, one hydrogen from here and this hydrogen from here, they are removed. So, you get it here as CO. So, this is a dehydrogenase reaction. The process of mitochondrial fatty acid oxidation is termed beta oxidation, since it occurs through the sequential removal of 2 carbon units by oxidation at the beta carbon position of the fatty acyl coenzyme molecule. Each round of beta oxidation involves 4 steps that in order are oxidation, hydration, oxidation and cleavage. Once the fatty acids have been transported to the mitochondrial matrix via carnitine pathway, beta oxidation occurs within the mitochondria in 4 steps as discussed below. The first step, where fatty acyl coenzyme is acted upon by an enzyme acyl CoA dehydrogenase, which is FAD dependent. Fatty acyl coenzyme A undergoes dehydrogenation and forms a trans double bond at their alpha and beta carbons to form trans-enoyl coenzyme A. Acyl-CoA dehydrogenases are present as 3 isoenzymes, each specific for a particular carbon chain length. They are short, intermediate and long. Short chain acyl-CoA dehydrogenase prefers fatty acids of 4 to 6 carbons in length. Medium chain acyl-CoA dehydrogenases prefer fatty acids of 4 to 16 carbons in length with maximal activity for C10 acyl coenzyme. Long chain acyl coenzyme dehydrogenases prefers fatty acids of 6 to 16 carbons in length with maximal activity for C12 acyl CoA. The electrons which are removed from the fatty acyl coenzyme chain are transferred to FAD, which gets reduced to FADH2. This FADH2 immediately gets converted to ATP molecules via the electron transport system. Now, second step, enol CoA hydratase catalyzes this reaction where water is added. Hydration occurs at the double bond, resulting in the formation of beta hydroxy acyl coenzyme A. In the third step, beta hydroxy acyl CoA undergoes dehydrogenation to form beta keto acyl coenzyme A in the presence of beta hydroxy acyl CoA dehydrogenase. The electrons available as a result of dehydrogenation are accepted by NAD to form NADH, which immediately changes these electrons with oxygen in the electron transport system to form ATP molecules. Now, in the fourth step, this reaction is called as thiolysis, as acyl CoA acetyl transferase, which is also known as thiolase, in the presence of coenzyme A causes the cleavage of beta keto acyl CoA to form acetyl coenzyme A and the thioester of the original fatty acid with 2 carbons less. This cleavage occurs at the beta carbon ketone group is a good because it is a good target for nucleophilic attack by the thiol group of coenzyme A. This again participates in the beta oxidation cycle to form another new fatty acyl coenzyme A with 2 more carbons less and a new molecule of acetyl coenzyme A. This process continues till the entire fatty acid is converted to acetyl coenzyme A molecules. So, that is how it is completed, a round of beta oxidation is completed. Now, each round of beta oxidation produces 1 mole of FADH2, 1 mole of NADH and 1 mole of acetyl coenzyme A. The acetyl coenzyme A, the end product of each round of beta oxidation, then enters the TCA cycle for further oxidation. Now, coming to oxidation of odd carbon fatty acids, the majority of natural lipids contain an even number of carbon atoms. A small proportion of plant derived lipids contain odd numbers, and upon complete beta oxidation these yield acetyl coenzyme A units plus a single mole of propenyl coenzyme A. The propenyl coenzyme A is converted in an ATP dependent pathway to succinyl coenzyme A. The succinyl coenzyme A can then enter the TCA cycle for further oxidation. Now, coming to oxidation of unsaturated fatty acids, the oxidation of unsaturated fatty acids is essentially the same process as saturated fats, except when a double bond is encountered. In such a case, the bond is isomerized by a specific enol CoA isomerase and oxidation continues. In the case of linoleate, the presence of unsaturation at the 12th position results in the formation of dienol CoA during oxidation. This molecule is the substrate for an additional oxidizing enzyme, the NADPH requiring 2,4-dienol CoA reductase. Now, coming to the second aspect, biosynthesis of fatty acids. Fatty acid biosynthesis occurs in the cytoplasm, whereas fatty acid oxidation takes place in mitochondria. Fatty acid biosynthesis uses a 3-carbon intermediate called acyl carrier protein, which is in short called as ACP, instead of coenzyme, which is in short called as COA, as in the case of fatty acid oxidation. The reducing agent NADPH is used in fatty acid oxidation, whereas the oxidizing agents NAD and FAD are used in fatty acid oxidation. Moreover, the biosynthesis requires the participation of a 3-carbon intermediate malonyl-CoA that is not involved in fatty acid oxidation. Fatty acid biosynthesis can be divided into 3 separate pathways as shown below. Number 1, synthesis of palmitic acid from acetyl coenzyme A. Number 2, elongation of palmitic acid. Number 3, desaturation. Now, acetyl coenzyme A is produced in 2 ways in the mitochondria. One is by beta oxidation of fatty acids and 2 from pyruvic acid produced from glycolysis. Acetyl coenzyme A will accumulate when the oxidative phosphorylation slows down. Under these conditions, acetyl coenzyme A is transported out of the mitochondria to the cytosol, where it can be used in fatty acid synthesis. The first committed step of fatty acid biosynthesis is catalyzed by the enzyme acetyl-CoA carboxylase. The enzyme contains biotin and adds a carbon dioxide, resulting in a carboxyl group to the methyl end of acetyl coenzyme A, forming malonyl coenzyme A. This reaction is an energy requiring reaction. As you can see, acetyl coenzyme A in presence of a bicarbonate ion and the enzyme acetyl-CoA carboxylase and ATP molecule is converted to malonyl coenzyme A. So, this is the first step in fatty acid biosynthesis. As a first step, both the primer and extender substrates are attached to acyl carrier proteins. These are acetyl coenzyme A and malonyl coenzyme A molecules are then attached to acyl carrier proteins, which has the same prosthetic group as coenzyme A. A sequence of reactions follows in which the chain is extended and butanoate is formed. First, 3-oxobutanoate is produced by a reaction catalyzed by beta keto acyl ACP synthetase. Then this is reduced to 3-hydroxy butanoate by beta keto acyl ACP reductase, which is in turn dehydrated to trans 2-butanoate by beta hydroxy acyl ACP hydratase before it is reduced to butanoate by enoyl ACP reductase. The process then continues with the addition of a further 6 units of malonyl ACP by successive cycles of these reactions until palmitoyl ACP is formed. At this point, a thioesterase removes fatty acyl product as the free acid. We have just now discussed the reactions of fatty acid biosynthesis. Now, we will see the reactions here. We can see one molecule of acetyl ACP combining with one molecule of malonyl ACP to form beta oxobutanoate in presence of the enzyme keto acyl ACP synthetase, and one molecule of coenzyme A is released. Now, beta oxobutanoate is acted upon by beta keto acyl ACP reductase enzyme in presence of NADPH. So, NADP is released, NADPH provides 2 hydrogen atoms. So, it is converted to 3-hydroxy butanoate. 3-hydroxy butanoate is next acted upon by beta hydroxy acyl CoA ACP dehydrase enzyme, which removes one water molecule to form trans 2-butanoate, which is then acted upon by another enzyme enoyl ACP reductase in presence of NADPH2, where NADP is released, it is converted to butanoate. Now, this cycle continues by taking another 6 molecules of malonyl ACP to form a 16-carbon ACP unit. Now, 16-carbon ACP unit, once it is formed, is acted upon by thiol esterase enzyme. So, that ACP is now converted to coenzyme A derivative. So, 16-carbon ACP is converted to 16-carbon coenzyme A derivative. So, this is the palmitoyl coenzyme A molecule which is formed. The net reaction of the whole process of fatty acid biosynthesis is, we will start with one molecule of acetyl coenzyme A plus 7 molecules of malonyl coenzyme A 14 NADPH2 molecules, and we will get 1 molecule of palmitic acid, 7 molecules of carbon dioxide, 8 molecules of coenzyme A and 14 molecules of NADP along with 6 molecules of water. Now, next we will go into the elongation of the palmitic acid, which is synthesized by this process. The product of fatty acid synthase action, palmitic acid, is but of course one of many fatty acids synthesized by the cells. Elongases are the enzymes that act to lengthen palmitate to produce many of the other fatty acids. Elongases are present in mitochondria and the endoplasmic reticulum. Elongation using elongases in the mitochondria involves a mechanism that is essentially the reversal of beta oxidation, except the substitution of NADPH for FADH in the last reaction. Now, desaturation of fatty acids is the next process where terminal desaturases produce unsaturated fatty acids. One such enzyme is fatty acyl CoA desaturase. From the free methyl end, mammals cannot make double bonds closer to the end than the delta 9 position. Oleic acid is a delta 9 fatty acid. Thus, linoleic acid, which is a delta 9 and 12 double bonds acid, and linolenic acid, which is delta 9, 12 and 15 double bonds, must be provided in the diet of mammals, and these are called as essential fatty acids. When fat is broken down by the liver, glycerol and fatty acid molecules are released. The fatty acid is broken down further in the process of beta oxidation, and acetyl coenzyme A is formed as the end product. Few of these acetyl coenzyme A molecules which are formed are converted to a ketone body called acetoacetic acid. Acetoacetic acid is then converted into two other types of ketone bodies such as beta hydroxy butyric acid and acetone. Liver is the chief source of ketone body formation, and when the free acetoacetic acid once formed in the liver, it is not efficiently reconverted to acetoacetyl coenzyme A because liver is deficient in the activating enzyme. Hence, much of the free acetoacetic acid cannot be further metabolized in the liver, but passes into the circulation and is transported into the muscle and other peripheral tissues, which reconvert it into acetoacetyl coenzyme A. This is then split into acetyl coenzyme A and is utilized in TCA cycle, etc. Normally, the blood contains not more than a few milligram% of ketone bodies, and less than 0.1 gram is excreted in the urine per day, which means the rate at which the peripheral tissues utilize ketone bodies balances their rate of production in the liver. However, when the quantity of fatty acid metabolized is greatly increased due to starvation, very high fat diets or severe diabetes, the rate of ketone body production in the liver exceeds the capacity for their utilization in peripheral tissues. Under these conditions, the ketone body content of blood increases, which is called as ketonemia. Urinary excretion increases, which is called as ketonuria, and a condition of ketosis exists. If severe, the breath may smell strongly of acetone. Ketosis is abolished by increasing the metabolism of carbohydrates in the liver. In diabetes, this is accomplished by giving insulin, and in ketosis due to carbohydrate deprivation by giving glucose or substances readily convertible to glucose and glycogen. Untreated, ketosis may progress to ketoacidosis, coma and death. Ketosis is an energy state that body uses to provide an alternative fuel when glucose availability is low. It happens to all humans while fasting or when the carbohydrate intake is lowered. The process of creating ketones is a normal metabolic alternative designed to keep us alive if we go without food for long periods of time. Eating a diet low in carbohydrate and higher in fat enhances this process. The condition of ketosis typically represents a change in the way the body gets its energy. Normally, the body gets most of its energy by metabolizing glucose obtained from carbohydrates or stored in the liver and muscle as glycogen. But when unable to convert glucose into energy, the body switches to breaking down fat and converting it into energy. When this happens, the liver metabolizes fatty acids, producing energy rich ketone bodies. Now, to conclude, the major aspects of lipid metabolism are involved with fatty acid oxidation to produce energy or the synthesis of lipids. Lipid metabolism is closely connected to the metabolism of carbohydrates, which may be converted to fats. Fatty acid beta oxidation is the major metabolic pathway that is responsible for the mitochondrial breakdown of long chain acyl CoAs to acetyl CoAs. The acetyl CoA is then ultimately converted to ATP, carbon dioxide and water using the citric acid cycle and the electron transport chain. Fatty acids are synthesized from carbohydrates and occasionally from proteins. Actually, the carbohydrates and proteins are to be first catabolized into acetyl coenzyme A. Depending upon the energy requirements, the acetyl CoA enters the citric acid cycle or it is used to synthesize fatty acids. Ketosis is a metabolic state where most of the body's energy supply comes from ketone bodies in the blood, in contrast to a state of glycolysis where blood glucose provides most of the energy. Hope you have enjoyed this class. See you in the next session.